Thanks. So now I'll introduce our first speaker, Samuel Skibicki, who will be presenting a talk titled Phylogenomics and Biogeography of Xeniinae using plastome and nuclear data sets. Thanks so much.
Thanks, Colin, for the intro and for hosting this week. That is my title. He did mention that I'm now at the California Department of Food and Agriculture as the senior plant taxonomist. This presentation will be a condensed version of my thesis talk, and my thesis was when I was at West Virginia University. I defended this about a year ago. So how I got into the Xeniinae subtribe. It started by my master's work at Northern Arizona on San Vitalia, a member of the Xeniinae. So this was a really good master-sized project, and I did a full taxonomic treatment of the 6 species, a quick phylogenetic tree with ITS and 3 plastid loci, and a chromosome number analysis that resulted in some really cool-looking SEMs of the ray cypsele that wound up being diagnostic in my dichotomous key. We also found 4 occurrences of downward disploidy, you see with the black arrows on the tree on the right, and 5 instances of duplication events. So it's a pretty interesting group. There are some things to find, but generally when you do a thing like this, you wind up with more questions than you answer. So for my PhD, I just expanded this to the entirety of the Xeniinae using a lot more robust molecular tools. I would normally go into all of the genera in depth. We're just going to have to go really fast through the range maps here. So this is St. Vitalia, the group I worked on. If you got a keen eye, you can see my lanyard today was made by my friend. This is St. Vitalia on my lanyard. So that's my pet group. There's 6 species. It's got a really interesting amphi-equatorial disjunct in that we've got St. Vitalia versicolor in South America, and the bulk of the clade is in Mexico and southeastern U.S. Tijuana, really quick one. It's in Oaxaca and it's monotypic. As Trichocorony is also monotypic. It grows on the Nayarit, Sinaloa, and Durango tripoint in some vernal muddy pools there. Phylactis has 3 species in southern Mexico, mostly in Oaxaca as well. This is where we get into the ones that are generally known because they're into horticulture. Echinacea is a big one for sure, 9 to 10 species, depending on who you talk to. Really prevalent in the Great Plains of the United States. It's this grassland that runs right up the middle all the way into Canada. They do occur in the eastern US, but take the range map with a grain of salt because these are really into horticulture. So a lot of these can be plantings and they weren't natively occurring, but they do occur on the East Coast as well. Same thing for Heliopsis, where this does get into cultivation, but it is also found in North and South America, like we saw in San Vitalia. And then the big one, as you can see, there's the least amount of work that's been done on Zinnia because there's anywhere between 22 and 27 species, depending on who you ask, all across North and South America. But again, this doesn't get into cultivation. So we even see people planting Zinnia in Hawaii and in Europe and all over the world just because it's such a pretty one that's gotten into cultivation. I would like to quickly mention that the sister subtribe is the Spilantini, and that's been known from prior molecular work. So a good way to summarize my 3 chapters for my PhD thesis were just number 1 was plastomes, number 2 was the nuclear genome, and number 3 was biogeography. So we'll just jump right in to get started on those. In 1963, Andrew Torres took a morphological crack at breaking Zinnia into different subgenera, which he came up with Diplothrix and subgenus Zinnia. And the subgenus Zinnia was then further broken into 3 different sections, Mendisia and Tragoceras, and of course, section Zinnia. Mendisia and Tragoceras in the past have been their own genera, but in this case, Torres treats them as sections, which they seem pretty good. Zinnia looks to be monophyletic, but we'll see later on. And these colors will remain consistent as we see when we check the monophyly of these sections. So I don't have time to go through the aims, but for the questions for sure, we want to know is the subtribe Zinniini monophyletic? Are the genera inside Zinniini monophyletic? And are those subgeneric delimitations that Torres came up with monophyletic? I would be remiss if I didn't talk about herbariomics. This is a large part of all the work on Asteraceae that I've done. Just an exorbitant amount of money if you were trying to travel to all these field sites, and especially a lot of time. If you can visit herbaria and they have destructive sampling policies, you can get some really good DNA, especially when you're using the Illumina system. You can get really good DNA out of 70 and even 100-year-old specimens. They're what's called pre-sheared, as I call it, because the DNA does break down. If you were doing something like PacBio, you'd have trouble with older material, but I've had really good success with CTAB with some decades old specimens. So check in with your local herbaria about their destructive sampling if you'd like to do a project similar to these. A little bit about me is I now curate the CDA Herbarium here in Sacramento, California. We've got about 61,000 specimens. I have to skip methods, unfortunately, so let's just jump right into the fun stuff, to the results. We wound up finding that Heliopsis is paraphyletic. We've got the core down at the bottom in orange, and there are two species, Heliopsis longipes and Novogalaciana, New Galicia Heliopsis in Mexico. Those are the two. I'll probably refer to those as the oddballs. They come way out. So not only are they outside St. Vitalia, excuse me, not St. Vitalia, outside the Zinniini, they're outside the Spilantini subtribe as well while using plastome data. They're outside with Trichocorny. That one's always been considered an oddball in the Zinniini. And this is just a split that has to be dealt with, not a split, excuse me, a pattern of paraphyly that needs to be dealt with taxonomically, probably a new genus. Let's jump into Zinnia, which I showed you before with all those colors, making a tree with plastomes. You can see that in orange section Tragoceras comes out really nice, but the other ones are what you call messy. There's paraphyly all over the place here with our different sections, but we'll deal with this later when we've got nuclear data as well. And lastly, Zinniini itself is paraphyletic due to this Heliopsis coming outside of the Spilantini most recent common ancestor. So there's two ways to approach this problem. Is on the left, we can keep it as it was, where Zinniini with the blue star at that node there, and keep the Spilantini separate with the red star like they've always been, I think since about 2000. It was... The name is escaping me. It'll come to me eventually. I don't have time to dwell or tangents when I practice this talk. I was right at 15 minutes. So the way to do this on the left would be like a new subtribe, so that we'd have like a 3, possibly 4 species subtribe. There is another Heliopsis species that has been morphologically linked to Longipes and Novogalaciana. So there's probably a third oddball that I didn't get to add into this study. So we can have a really small subtribe. Or you can just, on the right here, just move the Zinniini to include all the Spilantini and the Trichocorny and the oddballs. You've got a really tight group. That's the way I propose to do this. It's not that Spilantini is not phylogenetically, this is a good, it's still a good clade. It's monophyletic. You can still have some utility to it, but it just gets swallowed up here by a broader sensu latu Zinniini. So for aims and questions, we gathered the data and we wound up making our trees and informed the taxonomic changes. One that came here, and the things inside the Zinniini, those sections, we'll deal with those later. But Zinniini was not monophyletic, but it is now with that new circumscription. Are the genera of Zinniini monophyletic? No, Heliopsis was not, so there's some work to do there. And inside Zinnia, we saw no, that those sections are not monophyletic. So jumping into chapter 2, again, with the aims, we'll have to skip those, but the questions are, if we do see gene tree discordance when using our nuclear data back to trees, will incomplete lineage sorting be able to explain all of the differences that we see, all this gene tree discordance that we see? And if not, can we detect hybridization? And also, did the subgeneric delimitations inside Zinnia show patterns of paraphyly from the nuclear perspective as well, as we saw from the plastomes earlier? So again, we'll have to skip the methods. If you have questions on those, feel free to email me later or ask me after this, but we've got to move on to this Astral Pro tree that gives us a topology with the most concordant patterns of our quartets. We've got 15 nodes that did get flagged by PhyTop as not falling with what we'd expect with incomplete lineage sorting alone. So what that looks like is with our blue bars here is the concordant quartet. And then Q2 and Q3, you'd expect to be at even numbers. And for most of the tree, the nodes were, but for these 15, you wind up with more than more of Q2 or more of Q3. And that means that there's something other than random chance going on when they're splitting in the deep past. So what we did was we ran Philo networks, which is a very large and computationally prohibitive thing to use if you have many, many tips on the tree. So we really had to pare this down to only 10 tips. We wound up finding that there's seems to be about one ancient hybridization event, and it comes out here between Heliopsis and Zinnia. I could do some more work on this and maybe try some different species and see if that works with different sections inside Zinnia. But for now, we've detected the one. It seems to be something that had happened deep in the past with hybridization. And also, those sections inside Zinnia did wind up being paraphyletic, so we're going to have to change those. We're going to need a taxonomic revision of Zinnia. So here's what that looks like. This was good practice for me, especially since I'm the senior plant taxonomist with CDFA now. Zinnia, just these 5 changes need to be made here with the blue stars, just changing these species into different sections, and we wind up with monophyletic sections after those changes. Torres, I think, did a pretty good job in the 60s, only using morphological data. So if he had molecular tools in his toolbox at the time, he probably would have come out with these. But kudos to him, did a great job on those papers from the 50s and the 60s. So for our aims, we did wind up gathering all the data we needed and making some really nice trees. And we informed whether those changes should occur. Incomplete lineage sorting could not fully explain all of the gene tree discordance that we were seeing, and hybridization was detected between Heliopsis and Zinnia. And again, Zinnia was shown to be paraphyletic, so we had to change those. For chapter 3, biogeography, questions that I was really interested in after we calibrate our time tree is what are the divergence times for the major clades inside Zinniani? Is Echinacea the most recently diverged? So if anyone has keyed anything in Zinniani before, I really enjoy the Sinvitalia and the Heliopsis and the Zinnia keys. There's a lot that are different, and you can really tell what was going on with the person who wrote the key about how they split these things apart. The Echinacea key I've always found to be very frustrating because the species themselves are very the divergence morphologically, it's barely there. You can really hardly tell what they're talking about in the keys, and the plants themselves do look very similar. So does that mean that they've just been around for less time? We'll find out. What is the center of origin for Zinniani? And do changing landscapes, in this case vicariance, lead to less, equal, or more speciation when compared to dispersal, when you're moving seeds elsewhere, and then you wind up with speciation later when they move habitats instead of the habitat itself shifting. So we wound up with the tree we used from chapter 2, and we put some dates on it. So we calibrated it with the bottom left corner is a fossil pollen grain from the Ambrosianae subtribe. And those are very distinctive, so they were able to place that into that group. And when you use the Ambrosianae as an outgroup, you can get a date on the outside of this tree. But for an internal calibration point, we used a paper from Zhang. I can't remember the year off the top of my head, but that paper used uncalibrated trees. They didn't have any fossil calibration that they could have used. Everything else in these nodes, as far as I can tell, there's no fossil evidence to calibrate these time trees off of. So I used theirs, which was not calibrated. So it's definitely something we'd have to go through and find some more calibration points. But this was the only thing I could use for an internal calibration, so I did have to use it. At every node here, you can see the blue line. That is our gap of where we think these splits would have taken place. So for an ancestral range estimation, we used RASP4, and we wound up with this tree, which I found very interesting. So for the bottom left, this very point here with the green arrow, we've got, this is the split between Spilanthaene and the Zinniani. And there's a 64% chance that the center of origin at this node was in Mexico. But if you go one node down to Zinniani sensu stricto, the original delimitation of Zinniani, We wound up with a 96.6% chance that this was a center of origin in Mexico. And here in blue in the middle, you can see where Echinacea becomes a US and Canada only um, as this diverged, which we'll get into right here. So here are some ages of those groups. Right off the bat, Zinnia is about 15 million years old. It seemed to diverge with these different sections between 14 and 10 and a half million years ago, which is really cool because you can imagine that why other researchers would have considered things like Mendesia and Tragoceras their own genera at different times because these are relatively old. When you see something like Echinacea, it's only 10 and a half million years old. And that is what we expected. There's such little divergence here, and they wind up being the youngest clade in the group. Further analysis with RAPS, with RASP, we wound up with Vicariance showing very little hits in RASP4, but for dispersal, we get many, many more peaks, and really interesting to find. I went back into some geological records, and you wind up with this thing called the LMCI, the late Miocene cooling interval. And this time, about 11 and a half million years ago, the Great Plains, this grassland in the central United States and Canada, was more of a savanna before that. But during this time, it became a grassland. And that is when the center of diversity in Ziniini came up from Mexico and wound up being dispersing to this brand new grassland as it shifted from a savanna to a grassland, and we get all kinds of diversification in this group. So here's a pretty picture from central United States where we've got echinacea growing in these grasslands. If anyone has visited any of these states, looks something like this. So this story, people are always looking for stories to tell in biogeography, and that seems to be the story of Ziniini. Is the center of diversity is in Mexico, and once this new grassland becomes available, echinacea moves up and diversifies. And now, whether there were other Ziniini species that were in there during when it was a savanna, that's the question I haven't answered, but something that keeps me up at night for sure. Maybe they all went extinct, or maybe they did and they came back and diversified into something else. But that's the story that I'm telling. So conclusions from here, we wound up making our chronogram with a really good deep calibration node using that fossil pollen from the ambrosia. We studied the biogeography of Ziniini, and the divergence times for the major clades were taken care of, which was cool. And we were correct in thinking that Echinacea is the most recently diverged lineage. The center of origin for Ziniini seems to be Mexico. And the changing landscapes lead to less, equal, or more speciation when compared to dispersal. It seems like dispersal is what really drives diversity in the Ziniini that peaks during the late Miocene cool interval. Future analyses that I'd like to get into, taxonomy of Heliopsis for sure. So I've only read all these papers. I haven't done a full loan and taken a really close morphological look myself at Heliopsis. Before I'd want to change any of that taxonomy, I'd like a morphological feature that splits that apart from the rest of Heliopsis, those oddballs. Everything I read about them, no one really seemed to notice that they were that strange. They were just put into Heliopsis. They seem like a good fit. But the molecular data say they aren't such a great fit. So we're going to need a new genus. But first, I'd like a morphological feature that pulls them out consistently. More biogeography that I'd like to get into would be arid environment adaptations that some of these plant have with smaller leaves, more plant hairs, things like that. Also, Heliopsis helianthoides has a species complex with 2 or 3 varieties, depending on who you talk to, with a species called Heliopsis gracilis that grows on the southern edge of the Heliopsis helianthoides range, and it might be more of a variety like these other ones. But I've got some preliminary molecular work on that, and that's been on the back burner for years now, but hopefully one day I'll get to tackle that one. In the meantime, here are my acknowledgments. Hopefully I didn't go over too much with time. I'll take any questions, and I appreciate your time listening.
Thanks so much, Sam. We have some time for questions, and oftentimes during TICA talks, we can collect some questions now and address others that might come up for speakers at the end as well if we have additional time. In this case, however, Sam is going to need to leave promptly at the end. So if you have questions for him, I hope you'll please raise them now. Yeah, Jen.
Sorry, I was trying to figure out how to raise my hand.
Yeah.
I'll just do it for real. Great talk, Sam. I love hearing your work. It's so exciting. I had a question about your really cool hybridization between Heliopsis and the Zinnia clades. I was trying to figure out how old you thought that was. And then is there some morphology around that, maybe some of those original authors might have seen? Were there any hints to that?
Yeah, so a lot of what is diagnostic in Zinniini are these really ornate ray Sipsley awns, and the two genera that are lacking, well, Echinacea doesn't have them as well, but Heliopsis and Zinnia are lacking them. Heliopsis sometimes has these smaller ones, but wasn't a surprise when I found that these were the two that had a hybridization event. I'd really have to like dive in and become. It took me 3 years to become the morphological expert after Torres's passing of San Vitalia. So I imagine becoming the expert in Zinnia's 25-ish species and uh, Heliopsis' 18 would take a maybe a lifetime or decades at least. So I don't know, but it wasn't too much surprise. They do seem a little bit similar, and I'd have to. Yeah, I want to kind of go back into phylo networks and play with some different tips and see if it comes
Yeah,
out section wise in Zinnia as well.
cool. Thanks.
We have a question from Mike.
Hey, thank you. A fantastic talk. I just had a real quick question. So you mentioned that you saw that there were some disploid changes as well as duplications in the background there in Zinnia. Do you see any association of those with these transitions in habitats or aligning with this new taxonomic structure that you're resolving with phylogeny? Thanks.
Yes, so San Vitalia, the two sections that I split off there, certainly did. There's one San Vitalia Birdii that is 11, while the others are either 8 or 16. So that was a really clean split. I haven't looked too closely at Zinnia or Heliopsis. Echinacea had one duplication in it, but it is kind of the new guy on the block. But yeah, I'd have to go back into Zinnia and Heliopsis to see if those splits did kind of hold up. Because I know that there are. I actually have a slide. I didn't include it here. Zinnia has like 8 or 9 different chromosome counts in the genus. So I'm sure there's definitely something going on with the sections there.
Cool. Thanks.
Well, thanks so much, Sam. We really appreciate your time. We should move on to Marissa. I'm now happy to introduce Marissa Schubert, who will be presenting a talk on methods for resolving paralogs and phylogenetic discordance in Heterotheca. Marissa?
Okay, great. Thank you. All right, let me. Okay, can everyone see my screen? Okay, great. All right, yes. So for those of you who don't know me, so I'm Marissa Schubert. I'm a fifth-year PhD candidate at the University of Oklahoma in Abby Moore's lab, and I'll be talking today about Heterotheca, which is an Asteriae. It's in the tribe with goldenrods, asters. If you're in North America, there should be lots of familiar genera and a lot of kind of DYCs, especially in the western part of the country. Heterotheca isn't immediately distinctive from a lot of other comps in this tribe. It has a very kind of typical generalist floral syndrome. And in the way that the genus isn't immediately distinctive from other genera at first glance, many species within Heterotheca aren't particularly distinguishable or distinctive at first glance until you get familiar with some of the relatively cryptic morphological features used to distinguish different species. Heterotheca does have some really interesting phenomena in it, though, including repeated origins of polyploidy within the genus. Hybridization has been perceived to occur very extensively, although part of this might be better attributed to high morphological variation within taxa. There have also been repeated niche shifts to rock outcrops to essentially areas with very little soil to reduce competition. To evaluate these and other factors, we wanted to first develop a robust phylogeny to evaluate the genus. The first challenge was trying to figure out kind of where and what to sample because the taxonomic treatments for the genus differ really drastically. One is 25 species with a whole ton of infraspecific taxa, and another has about 70 species with just a couple varieties in it. We took a fairly broad approach of including 95 total samples to cover just about every taxon in both treatment. We also included in this 95 are 9 outgroup genera. And a special thanks to the 17 herbaria, which managed to get us tissues for help with this genus-wide phylogeny. the sequencing we employed was the Asteraceae specific bait set that Jen Mandel's lab created. When we sequenced everything, we had really great locus recovery for individual samples. There were only really 2 or 3 samples that were missing a fair amount of data, but everything else had really great recovery. If we were to concatenate all of our alignments together, we get about 250,000 base pairs, and we even managed to get some off-target sequences like ribosomal and chloroplast data. The chloroplast data wound up not being terribly informative, but our nuclear ribosomal tree uncovered kind of 3 main clades within Heterotheca. They aren't a one-to-one kind of correspondence to the previous sectional treatments of Heterotheca, so we'll just be calling them blue, yellow, and red for simplicity today. If we look at the low copy nuclear or kind of on-target sequences that we recovered, we see these same 3 clades emerge, although I'll note that there's lots of dashed lines, especially in this red clade here, that indicate that there's relatively low or modest support values from Astral, which will be a reoccurring theme within this red clade here. Within our blue clade, though, of just a few taxa, these are predominantly kind of southwestern US or northwestern Mexican taxa that include both the only discoid species and also some with stipitate glands, a relatively unusual feature for a lot of the perennials. Our yellow clade comprises probably the most kind of economically significant taxa that include annuals and perennials that have kind of this heterocarpic reproductive strategy where some seeds will disperse immediately, go off and germinate the first year. Others will lie dormant for a few years before germinating to help kind of protect them from adverse conditions, as many of these are annuals. And then our largest and most diverse red clade, which is also the troublemakers, includes basically all the taxa which have transitioned to being lithophytes, essentially, where they'll grow out of basically just rock outcrops with very little amounts of soil for them to anchor themselves to, as a likely way of avoiding competition with other faster growing species. The features used to differentiate taxa in this red clade are also relatively subtle. They're often small features that rely on gland features, hair features, some kind of leaf dimensions, or the presence or absence of bracts on them, but the differences can be relatively cryptic at first viewing. This red clade is also the most geographically widespread and encompasses somewhere between 40 and 60 taxa, most likely. There are a few widespread species, like Heterotheca villosa in the strict sense, but most of them are relatively narrow endemic, like Heterotheca jonesii in southwest Utah. This is one of a few species that have become dwarfed, essentially, where there's only really just 2 or 3 inches to the entire plants, including their blooms and everything at full maturity. These encompass a wide breadth of habitats, but typically like arid or semi-arid prairies, meadows, rock outcrops. Even in the eastern US, where there's more consistent precipitation, we see that they tend to occur in really sandy, gravelly, or rocky soils where the water doesn't stagnate for too long. This is also by far the most complicated group in Heterotheca, where hybridization is believed to occur fairly rampantly across many taxa. This is also where all of the polyploids are located in Heterotheca. To try and figure out why the resolution in the red clade wasn't what we might have expected at first, I looked through essentially all of the alignments at kind of when they were first assembled, and we noticed there were hundreds of alignments that contained paralogs, essentially non-orthologous gene copies that were giving kind of conflicting signals. If we look at things through 5 parts, which is just a way of assessing concordance or discordance in them, we see that the majority of these nodes here, or the majority of these little slices of these pies, are red, indicating that discordance or kind of disagreement among the gene trees is what predominates here, rather than a lack of resolution. To try and figure out what's going on a little more, we employed something called concordance factors, which is just another way of assessing support on a per gene and a per site basis. So GCF, or gene concordance factors, look for essentially which genes support a given relationship, and SCF, or site concordance, look essentially at individual base pairs to see which are kind of decisive in supporting certain relationships. We see that the gene concordance factors overall are very, very low. Most of them for most nodes are under 10 or 15%. We see that the site concordance factors are kind of between 25 and 55%. So there is data here, but each gene only holds a little bit. We thought that paralogs might be responsible for this, so we tried to resolve the paralogs using a variety of different combinations of methods, first starting with different assembly methods. These included Hyde-Piper, which is kind of our standard assembly method. We also employed Hyde-Phaser, which essentially rearranges Hyde-Piper-derived loci to make supercontigs, then filter out various samples, loci, and sample locus combinations based on a variety of factors. We also incorporated OrthoFinder, which reshuffles amino acid or protein sequences rather than DNA sequences in a more complex way to also try and filter out paralogous or highly divergent sequences. What these generally led to were about 1,100 loci for each assembly method. For the loci which had all the samples represented, we had about 400 for Hyde-Piper and OrthoFinder. This number decreased for Hyde-Phaser just because of the way that samples are filtered out. And then there was also variation in how long individual alignments were. I'll note OrthoFinder is by far the shortest because the DNA gets translated to protein sequences, so it gets kind of divided by 3 that way. If we average the concordance factor values for all of the different assembly methods, tree inference methods, alignment methods, and whatnot, we do see that Hyde-Phaser, the red dots, do have slightly higher values than things like Hyde-Piper in green, or especially OrthoFinder in blue. But if we zoom out to kind of the full range of this chart, we see the gains that you get from changing inference method, alignment method, assembly method are really small in the grand scheme of things. This was a little bit unexpected at first, and so we wanted to try and investigate what other factors might be used to try and increase resolution or filter out paralogous loci. For this, we looked at some gene tree statistics, looking at 4 different kind of subsets of data within each of our assembly methods. All just refers to all of our loci. HP2 refers to those which Hyde-Piper automatically deemed non-paralogous. Viz are just ones that I went through alignment by alignment and essentially just made a list of which ones appeared to be paralogous and those which didn't. And then rep is just to account for missing data. Those are just our alignments that had all of our samples in there. When we filter or compare statistics across gene tree values, we see that bootstrap support values are basically the same within assembly methods and across our 4 kind of subsets of data. I'll note that while bootstrap support values are generally better when they're higher, you can also get some bad bootstrap support values where you get these very long branches in your trees that are results of essentially multiple paralog copies kind of filtering out. So on one half of the tree, you have the first paralog copy. On the other side of the tree, you get the other portion. And then the long branches in between are all the differences between the paralog copies. If we look at bipartition values, we see that there are some differences, especially for the Hyde-Piper-derived loci, but these differences are very small in the grand scheme of things. More interestingly, if we look at the internal branch length values, essentially how long the individual trees are, we see that the viz, or the manually curated set of loci, they are shorter on average. This, I think, is a good thing, and I'll explain why in a second here. I'll also note there are some outliers there that generally got filtered out in the manual version. If we have a gene that's basically just one big polytomy with just 1 or 2 SNPs, we see basically just this vertical line, and then there's just a couple samples that split off, but these aren't shared loci, so these genes are functionally uninformative. We have kind of the Goldilocks zone, or ones that have a fair amount of variation or SNPs in them, but which don't have different paralog copies. Conversely, we could have alignments like the tree we saw earlier, where there's one paralog copy that's kind of in the top right here, and another that's in the bottom left, and then those long branches with the red numbers are essentially the kind of differences between those 2 copies that you see. We tried to filter out loci that had these paralog copies as indicated by really long branch lengths. When we did that, though, for just our red clade here, That removed about 20% of the total available loci. We didn't see many changes to the overall topology, but what we largely observed was when you remove about this 20% of data, we essentially see just a decrease in support values among our data without really having a beneficial impact on the topology. We wanted to investigate other sources that might be making the data more complicated. To do that, we looked at possible reticulation events, especially within the red clade's evolutionary history. And I'll note that while we ran these hybridization analyses through SNAC or Philo networks, it was too intensive to try and run all 95 samples with all 1,000 plus loci, so we did it in pieces. For sake of time, we'll just look at one clade of primarily California species of Heterotheca up top here. There were 3 reticulation events noted. We'll ignore this first one near the base. But if we look at one feature, we see that this is between 2 varieties of what's called Heterotheca sessiliflora that are parapatric with one another, at least modern day. These have been noted to have morphological intermediates between these 2 varieties, so it makes sense this hybridization event would be observed. Likewise, with another variety, Belanderi, this one introgressed with what's called Camphrata and then Echioides. I'll note that these are also kind of parapatric or slightly sympatric with one another, and hybridization or presumed intermediates morphologically have also been noted in this group. So it seems like where we'd expect to find hybridization in some of these, we are observing it in the genetic data as well. As for where we go from here, there are some relationships in the red clade that remain difficult to resolve. This is probably due in part to hybridization early on in its diversification, and also probably because the group is very young. We expect that it can't really be more than 3 or 5 million years old as a genus, so things like this California clade we wouldn't expect to be more than 1 million years old. While removing paralogs does undoubtedly have benefits in a lot of data sets, in cases where there's very little resolution kind of as is, especially in these really young clades like the red clade of Heterotheca, a decrease in data leads to a decrease in resolution or support without giving us kind of the beneficial change to topology that we might expect in most instances. So if I have a take-home message, it's to really diligently explore confounding factors in genetic data, figure out where possible instances of hybridization would be noted in the literature, and also to try and identify primary sources of variation across a lot of different support metrics, try and better understand how the genetic data are being parsed. So with that, I have my contact information here, and there are a ton of people that I'd like to thank, including a ton of different landowners in the 17 herbaria that made this project possible. So with that, if you guys have any questions, I'd be happy to take them now.
Thanks so much, Marissa.
Yes, so any questions?
Jen?
Okay. So I haven't run it or we haven't run it yet, but I know Erica shared with me that there's maybe a newer version of Snack that might be able to expand sampling. And I just wonder if you've been thinking about that. I mean, we've had to do the same thing, like break up and taxon sampling changes things, and it's so hard to know what to do. So I'm just wondering, like, what you have any next steps on that or yeah.
Yes, so there is a bit of software called QSyn that was recently developed by someone actually at OU. We have been able to run that kind of preliminarily for basically the entire red clade, and we see something like 7 or 8 reticulation events within that group. Some of the values are relatively low for those, so we're still refining it and trying to figure out kind of where these reticulation events have been happening within that kind of troublesome group in Heterotheca. So we are currently working on it, but not quite done just yet.
Cool. It's a challenge.
Yep, yep.
Thanks. Other questions? Well, we can come back to questions if they come up for anybody over the course of the next presentation. I'm pleased to introduce Jeffrey Finch, who will present a talk titled Low Chromosome Numbers Are Associated with Arid Conditions and High UVB Radiation in Diploid Macarontherany. Take it away, Jeffrey.
Hello, thank you. Thanks for that introduction. Let's get this. Okay, so yeah, I've shortened my title a little bit, but I'll be talking about environmental drivers of chromosome number evolution in this subtribe, the Macarontherany. So my research largely focuses on chromosome number evolution in flowering plants. In this image, you can see the distribution of haploid chromosome numbers in angiosperms and animals in blue. And you can see that there's quite a bit of variability in chromosome number, but at the same time, there's this big peak right around n equal 9. So the majority of flowering plant species have somewhere around 9 to 12 chromosomes in their haploid set. And the pattern that we see in animals is actually really strikingly similar, where the majority have somewhere around 12 chromosomes. And this similarity is really interesting when you think about the fact that flowering plants, but not animals, experience a lot of whole genome duplication, right? So this figure shows the estimated number of whole genome duplications in any given angiosperm lineage. So if you trace from the root of the tree to the tip. So the typical angiosperm has experienced somewhere around 3 or 4 whole genome duplications in its evolutionary history. And if each one of these is doubling chromosome number, right, you would expect to see an abundance of species with chromosome numbers in the hundreds, but we don't see that, right? So something is driving chromosome numbers back down following these whole genome duplications. And if you zoom in on just the Asteraceae, you see a very similar pattern, right? Where the majority of species have around 9 chromosomes. And then you have this large secondary peak at 18, which is probably driven by a lot of polyploidy, whole genome duplications. And then you also have a pretty substantial number of species with kind of unusually low chromosome numbers. And those are the species that I focus on in my research. In order to really try to understand this question of what drives chromosome number reduction. So cytogenetically, the mechanism is what we call descending disploidy. We heard about that a little bit already today, where there are a few different specific mechanisms that can lead to single chromosome number reductions. But the key thing is just that the chromosome number is decreased by 1 without losing any significant gene content. But in terms of what is today I'm going to be exploring a little bit more about a potential deeper driver, right? Ultimate driver of chromosome number reduction. I'm specifically looking at this hypothesis from Stebbins and Grant that low chromosome numbers may be adaptive in obligate outcrossing annuals in unstable habitats because of its effect of reducing rates of recombination. So the underlying logic of this hypothesis is basically that in an unstable environment, annuals are going to be subject to frequent local extinction, which then gives rise to them frequently dispersing to disturbed habitats and having to kind of recolonize those disturbed habitats. So if we think about a species with a sort of a typical angiosperm chromosome number of 9, natural selection is going to build up adaptive allele combinations in that individual, right, in that population. If a high chromosome number individual then colonizes a disturbed habitat, due to recombination, a lot of new maladaptive allele combinations will be introduced. So the idea is that this will kind of limit population growth. Whereas if we have a low number species, let's say n equal 2, it's not going to suffer as much recombination load, right? The fit allele combinations will be retained, which will facilitate a more rapid population expansion in those disturbed habitats. This is the idea. So to test this hypothesis, I studied this subtribe called the Macarontherany, which is in the Asteraceae, which is notable for its low chromosome numbers. So in this figure on the right, you can see that the base numbers of all of the genera in this subtribe are 6 or lower. These other peaks are likely polyploids. So this is really representing the left side of that distribution beneath the typical number of 9. And we even have this one remarkable species, Xanthosma grassley, that has only 2 chromosomes, which is the lowest that's been recorded in any flowering plant. This group is also primarily distributed in arid and semi-arid regions of the Southwest North America. So unstable habitats, right, where things like drought, flooding, wildfires can result in these frequent local extinctions. And the group also has a really unusually high proportion of annuals at about 28%. So it's really well suited to testing this hypothesis. Now, some of my previous research has established that the Macrantherinae experienced an acceleration of descending disploidy with their origin relative to sort of a background group of Asteridae. So that's what's shown here in these green bars, where you can see in the background group, the rate of descending disploidy is very low. And then with the origin of the Macrantherinae, we see this big jump. So those low numbers that we see definitely are resulting, at least in part, from rapid descending disploidy. And that rapid disploidy is also associated with the high proportion of annuals. So I found that the rate of descending disploidy is about an order of magnitude higher in annuals than in perennials in this group. So the work that I'm focusing on today is more focused on testing whether the low numbers in the group are associated with those habitats, those unstable habitats. Which we can depict that relationship very simply in this way, right? Habitat has an arrow leading to chromosome number, meaning causal relationship. But the hypothesis and some of my previous results also suggest there's a relationship between growth habit and chromosome number as well. So annual habit associated with low numbers. However, annuals are also associated with unstable environments, right? So in this figure, you can see the proportion of annuals among herbs, where a high proportion is shown in darker colors. And if we focus on this region that is where the Macrantherinae are concentrated, we have a high proportion of annuals. So we have to account for this relationship between growth habit and habitat that is independent of chromosome number as well. I also wanted to explore a role for UV radiation. So this is not really part of the original hypothesis, but we do know that UV radiation is a classigen, right? It can induce structural mutations, such as the kind that could potentially lead to disploid changes. And it's also elevated in arid and desert climates. So the idea is that maybe the habitat increased exposure to UV radiation is resulting in an increase in rates of chromosome number evolution, like that accelerated descending disploidy that I showed. And this could then facilitate a response of chromosome number to selection, right? Because if low numbers are favored, the species still need a way to respond to that selection. So how can we determine the relative importance of growth habit and habitat stability and UVB radiation on chromosome number while also incorporating phylogeny? Answer phylogenetic confirmatory path analysis. So this is the approach that I use to test the relative importance of these variables. So in this approach, you can test the fit of multiple causal models, each represented by directed acyclic graphs, like the graphs that I was just showing. And then the approach is PGLS to test for relationships between those variables while also accounting for phylogeny, essentially. So this shows kind of the underlying conceptual framework of the models that I tested, where annual growth habit is going to increase rates of recombination due to shorter generation time, while unstable habitat, according to the hypothesis, will select for reduced recombination. And together, that should result in selection for reduced chromosome number. While UVB radiation will presumably increase the rates of disploidy, which will then result in, again, decreased chromosome number. But we don't have direct data on rates of recombination in all these taxa or like branch-specific rates of descending disploidy. So we kind of have to skip that middle tier and just directly test for effects of growth habit, habitat stability, and UVB radiation on chromosome number, while also accounting for the interrelationships between those variables. Right. So as measures of habitat stability, I used three variables: aridity index, and then interannual temperature variation and interannual precipitation variation. So aridity index just comes from Trabucco and Zomer. This is a little bit counterintuitive in that a higher index means less arid conditions. You can see precipitations on the numerator here. So arid regions like the deserts in the Southwest have a low aridity index. So in other words, a low index is associated with a low stability. And so we would also predict that we'd find lower chromosome numbers in areas with lower aridity index. Whereas the interannual temperature and precipitation variations are a little more intuitive. Higher variation indicates a less stable habitat, right? There's a less predictable temperature regime. And so we would expect lower chromosome numbers in those highly variable environments for both temperature variation and same relationships for precipitation variation. And then for UVB index, I took that from the GLOVE database as annual mean UVB irradiance. So this depicts the all of the relationships that I tested for within my path analyses, where in the green box at the top, you have the three stability variables, and we're just kind of accounting for collinearity between those three. Purple shows UVB, yellow is growth habit, and then pink is diploid chromosome number, right? So we're testing for direct relationships between stability and chromosome number, as well as UVB and habit. Where the idea is if you kind of toggle on and off each of those relationships and test the fit of all of the possible models that you can explore that way, this results in 64 models, and the output is essentially a ranking of the best fitting models. And so the models vary depending on whether there's direct effects of stability, UVB, and growth habit, or not, right? I use the PhyloPath package in R to conduct the path analyses, where each model is assigned a CICC score, which is similar to the AICC score, and a p-value, where the models with the lowest CICC score are considered the best, and then you take the average of any models that are within 2 delta CICC of that. So you take the best fitting models and average them. Now I fit models separately so this here on the right is the tree that I used. Sort of in the top right are the Macroantherae. Their closest relatives are the Eurybioids, and then I included several members of the Asterae that are just closely related to the Macroantherae to sort of explore that whole part of the tree. And then I fit models separately to the whole data set, all of the Asterae, just the diploid Asterae, then all of the Macroantherae, and then just the diploid Macroantherae. Right now I'm just going to focus on the results of the diploid analyses because they're the most interesting. So this is the average model, or the average of the best models for the diploid Asterae. And so the only predictor that comes out as having a significant effect on diploid chromosome number is aridity index, but it is in the expected direction. It is a positive relationship. So higher numbers are associated with higher aridity index, which is less arid habitat. But so there's no significant effect of growth habit or UVB radiation. We look at all of the diploid Asterae in that tree, or interannual precipitation or temperature variation. When I repeat this analysis with just the diploid Macroantherae, so this part of the tree, we have a little bit more going on. So the relationship between aridity index and diploid chromosome number is still there. It's still positive, and it's actually increased in strength. And we also now see an effect of UVB radiation on diploid chromosome number. And this is in the predicted uh, direction. So higher UVB radiation is associated with lower chromosome numbers. Yeah. And then we also have this relationship between growth habit and interannual temperature variation, which I won't get into. So what we found was that in diploids, low chromosome number is associated with aridity, and when looking just at the macroantherne, also significantly associated with UVB radiation. This is consistent with the predictions of that instability hypothesis from Grant and Stubbins. I didn't show again, I just focused on the analyses of the diploids only, but this effect only results when we exclude polyploids from the analysis. So this effect is in diploids only, which is consistent with an association with recombination. And then this association with UVB radiation is a novel result. However, I'm really interested in this potential relationship between UVB and chromosome number, and I think that the ultimate drivers of that relationship are still unclear and warrant a lot more exploration. So it could be, my suggestion here is that the increased UVB radiation is resulting in increased rates of descending disploidy, which is just facilitating an adaptive response to selection based on arid habitats, right? The aridity of the habitats. But it's also possible that low number is conferring resistance to UVB stress itself, right? That stress on meiotic or mitotic machinery is kind of more easily tolerated if you have low chromosome numbers. So the ultimate drivers of this relationship are unclear, and I think would be very interesting to explore further. That's what I have. Thanks especially to Mike Barker, my PhD advisor, who's here today, for supporting me in this, and I will answer any questions.
Thanks so much, Jeffrey. And if you'll unshare your screen, there we go. Thank you. Yeah, do we have any questions? Jen?
I'm scared to ask this one because I'm not gonna sound as smart as I wish I were, but I'm curious about like the recombination and polyploids and diploids and that sort of the potential for that evolving, within a clade, like recombination rates and things. I know you mentioned, that's sort of like a black box piece, but I just wonder if you maybe speak a little bit more about that and just kind of the connections there.
Sure. Yeah. So we don't necessarily expect rates of recombination to be evolving very quickly. But the um, that sort of represents a future direction, right? Is to actually reconstruct the recombination landscapes of species in this group with varying chromosome numbers and see and test that assumption that chromosome number change is dramatically changing the rates of recombination or the recombination landscape. Um, but yeah, this also relates to why I excluded the polyploids from certain parts of the analyses because it's easy to understand, I think, when how two chromosomes fusing into one would alter rates of recombination in theory. But when you talk about the whole genome doubling, that's going to have a very different effect on recombination. And that sort of falls outside of the scope of that instability hypothesis. So it actually makes sense that the diploids only analyses support the hypothesis more than the analyses that include polyploids because the polyploids are essentially just going to introduce a signal that will confuse everything that has nothing to do with the hypothesis.
In general, they do. That's right. Cool. Thank you.
Thanks, Luke.
Yeah, thank you for the excellent analysis. And I would like to ask you, there is another group in the Asterae in Australia that sees the same type of decrease in chromosome number, which is brachyscome. You go from 9 to 2 chromosomes.
Hmm.
And it's an arid landscape. I don't know whether you have thought of that as another experimental setup, maybe in collaboration with some of our colleagues up there.
Yeah, yes, absolutely. So that's, unfortunately very far away, right? That group is in
Yeah.
Australia. So, I'm working with what's nearby. But a lot of this is inspired by work that's been done in that group by like Watanabe in the 90s,
Yeah.
where they found an association between low numbers and annual habit, and are testing this, exact hypothesis. So it is absolutely a great system to look at for future exploration of this hypothesis.
Because it would provide you an independent test on your mechanism and so on at the same time. It's really fascinating. Thank you.
Yes, yeah. Thank you for that question. Yeah, exactly. And Mike says in the chat, yes, heading there this year. So he's working on plans to expand into that clade.
Stay tuned. Hope to get some of that those genomes and things sequenced this year or next, depending on how the weather goes down there.
Yeah.
Thanks so much. Well, it looks like we're just about out of time. So unless there is a quick question or comment from the audience, we'll just have to leave it there. I want to thank our speakers and thanks to all of you for joining us for another TICA Talks webinar. We'll see you next time.
Thank you.